And for giving me the opportunity to tell you about what we've been doing and where we're going. It's kind of a special time for us, so it's time to really reflect on the past and some of our recent achievements, and then at the road ahead. Although after that introduction, maybe I don't need to talk at all. So when I looked at preparing this talk, I thought that the theme of a lot of what we're doing is precisely kind of in the title, is moving from very simple building blocks and seeing how you can get some really unexpected behavior, it's like the emergent complexity in for example, the system that we're studying, the cytoskeleton. And so I'm depicting some of these building blocks here, and I'll tell you a little bit more about them as we'll go along. And as far as complexity, I think we'll all agree that this is some sort of a very complex blob of interesting stuff, and I will tell you what that is in a little bit later in the talk. But I think we can all agree that that's pretty complex. So let me start simple, though. Let me start from the very simplest view of how things work in eukaryotic cells. In a lot of cells, this is an appropriate kind of zeroth order, let's say, diagram of what's happening. So you have a nucleus where you have the DNA and the associated machinery which processes the DNA, and it holds information about how to make stuff in the cell. And then that's basically, in some ways, could be thought of as the brain. And then you have near the nucleus an organelle called the organizing center. And from that organelle fans out an array of filaments called microtubules, which go from roughly this area out to the periphery of the cell. So from roughly the center of the cell to the periphery. And what's interesting about these microtubules is that they're very long. People who are working with nanowires will appreciate the fact that the diameter of these things is about 25 nanometers, and the length can be 100 micron. Okay, so the aspect ratio is just off the charts. And they're pretty robust, and they're polymers of the individual subunits, which are only 8 nanometers long. So that's a very interesting subsystem in its own right as a material system. But in cells, and so if you look at this structure, you can see kind of immediately visually that there is a reason why microtubules are part of the cytoskeleton. It really does look like a skeleton in a cell. And indeed, microtubules permeate the cell and help maintain cell shape. So that's part of their function. And of course, there are other filaments in a cell, most notably actin filaments, which also help maintain cell shape. So it's a complicated, more complicated story, but we're trying to keep it really simple. And the other role for microtubules is to support moving stuff around, like Saviz mentioned. So in a cell, it's really essential, especially in big eukaryotic cells, it's really essential to move things around efficiently, because a lot of individual functions are compartmentalized in these big cells. So if you want to degrade something, there is an area where stuff gets degraded. If you want to make chemicals like ATP that serve as essentially fuel in a cell, that's compartmentalized. If you want to work on DNA, that's compartmentalized in the nucleus. So when you have these compartmentalized functions, you need to connect them efficiently so stuff moves around so the whole cell can function well. And the microtubules serve as a basis for long-distance transport, for moving stuff around, and that's kind of shown schematically here, where you have different cargoes that are moving along these filaments. And one of the things that I haven't mentioned yet is that these filaments, they're made out of individual subunits, but these subunits are asymmetric. And if you stick asymmetric subunits in the same orientation one after another, one end of this filament is always going to be different from the other. So usually biologists designate one end as the minus end and the other one as the plus end. It's not electrical, it's just a designation. And but because the filaments provide the binding potential which is asymmetric, there are proteins that can bind to these filaments and then move along them, and we call these molecular motors. And because of the asymmetric binding potential, they bind in a specific orientation. So they land in one way, and then they have a structure such that when they get a molecule of fuel in, they move in a pre-programmed way, essentially. So the upshot is that these motors are pretty dumb. When they bind to these filaments, they move in one direction usually. They're usually unidirectional. So the motors that move cargos towards the nucleus are usually of the dynein variety. They move towards the minus end. And the motors that move cargos to the periphery are usually of the Kinesin variety. And that's your usual kind of dichotomy of transport, where one set of motors can fight another set of motors. And from a physicist's perspective, one of the most interesting things about this also is that these motors work step by step. Right, 8 nanometers is the periodicity, so they just work step by step. So they take 8 nanometer steps typically, and they can produce force on the order of a few piconewtons, maybe 1 piconewton. So when you think about the energy scale associated with the work being done per step, it's not that far from kT. So these motors work against a lot of noise, just Brownian noise, stuff bumping in them, all kinds of perturbations, and nevertheless they work reliably, and they add up to a reliable picture of transport in cells. So this is a very interesting system no matter how you look at it. And already we're getting glimpses that it's more complicated than this simple picture. Okay. So I'm going to tell you a couple of stories where we have explored how this simple picture evolves into something more complex. The first story I'm going to tell you is concerns the differences between these motors. Okay, so this is supposed to be a cartoonish representation of Kinesin motors, and this is for dynein. And already looking at this picture, several things are worth pointing out. First of all, cellular cargos, things that move in cells, are quite likely, a lot of them have many motors on them. It's not like they hop on one motor and they go. So that's the first piece of complexity I want to throw in. That you don't have just one motor moving things around. You can have multiple. And now, suddenly this...
Do they do those in turn? Or I mean, if I think of several different motors, I mean, are they competing with each other?
That's a great question. And generally speaking, you know, both are possible. So it's a long-standing debate in the field of whether there's a tug of war where they're just engaging against each other. For example, Kinesin versus dynein. So one is pre-programmed to go one direction, the other in the other direction, right? So there's a possibility of a tug of war, and that certainly happens. But the other possibility is that there's some regulator that turns one on, the other one off. And there are suspicions that we're starting to understand how that might work. There is machinery that binds to both Kinesin and dynein and potentially can turn one on and one off, kind of switch between these modes. So both of these, there's evidence for both of these. Probably both of these happen. Okay? The simplest case is certainly just a tug of war, where they all simultaneously engage and try to pull in their direction. So that's one thing, is that one of the points I wanted to make is that these motors can fight each other quite directly. And the other point is, just looking at these, you can see that they're very, very different motors. It's a cartoon, but it's not that far from how we think these things look. Now, I've developed a class on molecular motors, which I teach every once in a while. And so I go into gory detail about the structure of these motors and what happens when the fuel actually binds and how it rearranges itself. I'm not going to do that here, but and so I don't expect you to follow these diagrams, but it's a very convenient place to point out one big difference between these motors. For Kinesin motors, there is one place where the fuel molecule binds. So the gas tank is basically one molecule. For dynein, nominally there are 6 places, but 2 of them are not working right. Evolution got rid of them. So there are 4 places where ATP molecules can bind. So right away, you see the gas tank is 4 molecules versus 1 molecule. That's a huge difference before we get into any architecture differences. Just dynein is a much bigger, much more complicated motor. They come from different enzymatic families, just very little in common other than they work on ATP. And yet, if you look at how they perform, let's say you just reconstituted microtubules in vitro, you put on a glass slide, you let a motor run along, actually they're going to run at about the same velocity. Dynein is a little bit faster at room temperature, but basically very comparable velocities. If you look at force production, how strong these motors are, Kinesin typically produces, Kinesin 1 typically produces about 5 piconewtons of force versus the dynein we and others have reported is about 1 piconewton. But and this has been a very big controversy why there's this huge discrepancy. It's been shown recently that if you add a cofactor big D and also dynactin, so that's a complicated story there, then the top force becomes 4 piconewtons. And the Yildiz lab recently showed that if you actually take two motors and you pit them against each other, you figure out a way to hook them up and let them battle each other, they battle each other pretty effectively. So they're pretty well matched up, actually. So it looks like in terms of force, they're quite matched up. In terms of velocity, they're quite matched up. So in terms of, and processivity, I'm not showing, but they're actually on the same order of magnitude as well. So all the key parameters for these motors, from a functional perspective, they look very similar. And so the question is, are there any differences? And ultimately, that affects how we think about this transport. If you think about motors fighting each other, it's really critical to understand where the differences are, because any difference will immediately affect how we think of cargoes moving in cells. And so we decided to do what physicists usually do, to prod these motors, to take them a little bit out of their comfort zone. So we decided to examine what they do as a function of temperature. And the reason for that is, I already mentioned, even their gas tank looks different. So if you think about fuel consumption, just the energetics of stepping, temperature would be probably the first variable you would want to play around with to see if there are any differences. And what we were looking for is whether or not dynein motility is somehow different, moves differently from kinesin. And because it has 4 binding sites for ATP, whether we would see any evidence for that, any signatures of that. And ultimately, the other thing is, as I mentioned, that the energetics of stepping is not that far removed from KT, which means that there was a potential that there would be 4 differences in force production if you go to low temperature. So these motors might do something very interesting with temperature. So before I launch into that, I just wanted to briefly mention something that comes up a lot in physical chemistry, where basically if you think of an enzyme, in the simplest case, is basically a chemical reaction type situation where you have a starting point and the end point, and both are fairly stable, so they're energy minima, so that means there's an energy barrier in between, and the chances of you hopping over that energy barrier are determined by the height of that energy barrier relative to KT, so you get a Boltzmann factor, which basically determines how your enzymatic rate changes with temperature. And so we looked at kinesin versus dynein, and so this is velocity of these motors. So basically, you can see immediately that kinesin velocity over a wide temperature range fits well to a single Arrhenius curve. Actually, it does something very interesting at very, very low temperatures, but for biologically relevant range, it's basically a single Arrhenius curve. Whereas dynein data did not fit a single curve, and in fact, was distinctly 2 curves. Now, one thing I don't like about linear plots is the fact that when stuff grows really fast, like an exponential, stuff that happens at the bottom of the scale, even though it's significant, it gets scrunched together. So the way to get around that is to look at this formula again. And what you can do is you can take a log of both sides and plot the logarithm of the rate versus 1 over temperature. And you should get a linear relationship. So that's the Arrhenius plot, and that's what's shown here. And you can see that the kinesin data, shown in gray, fits a straight line really nicely. So it's a single Arrhenius curve. Versus dynein, which you can immediately see has a single break at about 15 degrees. So that was very interesting to us. And so we were quite excited about it, but we wanted to know what other properties are doing. And you can see here, so the processivity for kinesin, processivity refers to how many steps a motor takes before it falls off the filament. And that doesn't really change much with temperature. It declines a little bit. For dynein, it's about the same as at room temperature. The force production, we were hoping that we would see some major differences. Those we couldn't resolve within our error bars. There was no major differences. But so the major difference, by the looks of it, was the difference in velocity. The fact that below 15 degrees, dynein essentially very quickly dies. So this graph has numbers on it and so forth, but to give you a feel for it, essentially kinesin is pretty happy below 15 degrees running around, whereas dynein, very quickly, the activation energy here is about 3 times the activation energy here. So it's very quickly, by about 13 degrees, it's really hard to see any motion.
Could there be a similar process for kinesin, only at a much higher, much lower temperature?
Yeah, we actually think that that is happening for kinesin for entirely different reasons. For dynein, again, I don't want to go into the structural reasons, but there are 4 binding sites, and the AAA1, the first binding site, is the major one that's responsible for stepping, and that's what's determining the rate at high temperatures. But it's been shown recently that AAA3 is essentially a regulator, a switch. And what we think is happening is that that domain gets switched, and you basically turn off dynein below 15 degrees. Basically, the switch gets flipped based on the occupancy of ATP there. So what we're thinking is that basically, because dynein has multiple ATP binding domains, when you cool it down below a certain temperature, one of the domains gets rearranged and flips the whole motor off. So all of that is...
For context, back to that figure, they're within the temperature is corresponding to like a mammal's temperature.
Yeah, that's what I was going to go to next. So basically, if you think about what happens in mammals, we all exist at like 37 degrees, right? So, and if you think about hypothermia, induced hypothermia or something, that's down to like 29 degrees. So the question is kind of, how is this biologically relevant? This is interesting from the point of view of the structure of the motors, but what is the biological relevance? And we went looking, actually after we got this data, because initially we wanted to know what the differences would be. We were looking at actually the differences in structure between kinesin and dynein, so we were pretty happy with that information. But then we looked at the biology of it, and it turns out that there's a lot of papers, especially from the 70s when they were doing these measurements quite a lot, on what happens in mammalian cells now as a function of temperature. And so this is for rabbit and also for cat nerves, and in those in those cells, as you go below about 13 degrees, it turns out transport just stops. So what they were doing here is they were injecting these nerves with some radioactive compound, and they were looking at the wave of radioactivity going out. Okay, so they didn't have fluorescence back then, they didn't have fancy video back then, so they were looking at radioactivity. So they saw there was wave of radioactivity going out, and they saw that below 13 degrees, transport was dying. So this is for rabbits and cats. This is for rats and ground squirrels, the same. So basically, as you look across the mammalian species, you see that basically there's a cold block at about 13 degrees. And there's been quite a bit of work in the early 80s to figure out whether this is due to some other factors. How do you pin it down? What's going on? And the first thought would be maybe these microtubules fall apart at low temperature. And it turns out that there's a slight decline, but there's nothing dramatic happening at 13 degrees. It seems like a big mystery, and we think that we have a clue as to what's actually going on, because we have something that actually falls apart exactly at the right temperature. And so what we think is happening is that when temperature drops below critical temperature,
I'm still confused by what did it mean it was done at 13 degrees Celsius? Like what's
yeah. So you're in some cases, you take a nerve and it's technically still part of the animal, but usually you're looking at a cell culture. Usually you're looking at the individual cells. But in the case of ground squirrels specifically, it's a very interesting species because it's hibernating. And hibernating mammals get exposed to very low temperatures. So if you're asking specifically in which mammals this gets realized, these are probably your prototype where you're learning a lot about how do these things survive going down to like 5 degrees.
Okay, so their body temperature can actually drop.
Yeah, so there are mammals for which this is immediately relevant. And there are, if you're a biologist though, there's a relevance to, a lot of cell culture is being done with cells that have been frozen and unfrozen and so forth. So the mammalian cells get exposed to these temperature regimes a lot, and some mammals directly get exposed to these temperature regimes. So this is very relevant to all kinds of biology.
So there's the break in the, I guess the Kinesin thing, what was that?
Dynein.
Right? Does that occur in a variety of different animals, and if so, does it occur at about the same temperature?
So this is in mammals, like I showed. There's a few that have been measured, and it seems like it occurs about the same temperature. But outside of the mammalian species, then it varies, and that's where one of the things where, yeah. Well, so in bullfrogs, for example, like it was quoted in that paper, it's about 10 degrees. So what we think is happening is that when temperature drops below some critical temperature, dynein shuts down, and the cell detects that. If you blanket shut down dynein by other means, by chemical means, you can do that. And the cell says, oh, nothing is moving towards the minus end. Oh, wait, I'm going to start shutting down kinesin because I want to have balance in the fluxes. And so there is some regulation that kicks in and on the scale of a few minutes, and then kinesin shuts down too. And we think basically that shuts down all transport in cells below a certain temperature. So exactly the question that you're asked, then the sanity check is what happens in other non-mammalian species. And you need something that is an established system for dynein research. And of course, there was one which was very prominent, and that is yeast. There's been a long history of comparison between yeast dynein and mammalian dynein. So we were wondering what happens in yeast, because in yeast there is no cold block at all. There is no cold block. This transport doesn't stop at low temperature. Of course, as you get closer to 5 degrees Celsius, microtubules do fall apart, and then you don't expect any transport anyways. But there's nothing happening at 13 degrees or 10 degrees or something like that, as far as transport is concerned. So we wanted to know what happens in yeast. So we went to one of the major labs that works on yeast. We went to Ron Vale at UCSF, and we asked for their constructs for yeast, and they sent us protein, and we examined it. This is for Saccharomyces cerevisiae, and there is a break in dynein velocity for yeast, but this break is happening at about 7 degrees. In other words, basically you wouldn't expect any cold block there, because by that temperature, all the microtubules are gone anyways. So there is no real way to look. And dynein is also non-essential in yeast, so you'd have to look very hard to see signatures of a cold block in yeast. But certainly you don't expect anything, because by the time there's any change in dynein, then basically all the microtubules are gone anyway. So biologically speaking, here in yeast, dynein has the dynamic range across the entire relevant temperature range for its existence. Well, actually, it's plotted slightly differently. They're very comparable, actually. But it's interesting that this break occurs at very different temperatures, which suggests that dynein, even though it's very similar motors, the yeast dynein has been used as a model to figure out the structure of dynein in general. Despite the similarities, it looks like some fine differences can be used to tune the dynamic range. The dynein can be adjusted to suit the temperature range of the organism. So it's a very interesting finding in that sense. And the other interesting thing is that dynein and kinesin track each other so well over a wide temperature range, which is relevant to mammals. Let's not forget that suggests that there's a real evolutionary benefit to having a good match between kinesin and dynein. It's not just an accident that happened at room temperature. It happens somehow it's beneficial to the organism. So that gives us insight into the function of these proteins and ultimately into how to model transport. It's really essential to know that these match up really well if we want to understand how they're pulling against each other as an ensemble. So that's one side of the story. But if you're a physicist or a bioengineer or something like that, you might ask, what would happen if the regulation were absent? Because if you were doing this in a glass slide, or if you're doing nanotechnology, which is biomimetic, you were moving stuff with kinesin and dynein motors, you might not put into your system any kind of regulation. The question is, what would happen then? Or, which is what we were very interested in, what happens if you drop the temperature on a cell really, really fast? Because then the chemical regulation won't have time to kick in. That's a few minutes. So you'd have a window to look at what happens to transport, kind of breaking it apart. A typical physics approach to things. We don't understand it. Let's hit it with a hammer. Let's see what happens. So temperature is a good hammer. And we're trying to develop this into a very broad understanding in the community that temperature is a good hammer to use to study things that are otherwise inaccessible. Because how do you figure out what's happening in a live cell right under your microscope? But if you drop temperature, you have a probe of what's going on with your ensembles of motors. And you can study activity, which is very key. But, of course, we are kind of limited. And in space, maybe with the Crocker Center, we'll be more able to do more experiments. But we can certainly do quite a lot of simulations to try to figure out what would happen if you pit kinesin versus dynein motors. And you can see here that this is a typical case where what you have is at room temperature or even a little bit below room temperature, dynein can be set up to win. You can just make an ensemble of motors where there's a lot of dyneins, a few kinesins, and then dynein will win. But as you drop the temperature, what happens is that dynein turns off, of course, and then kinesin starts winning by default. And then the entire direction of motion changes from minus n directed motion to plus n directed motion. So this is really exciting because this means that temperature can be used as a regulator of directionality of transport. Okay, and let me show you a few videos. So this is what happens. So here, this is the results of simulations. So this is where the cargo is. These red dots represent the heads of dynein, and the green one, you can probably see it better here, is the lone kinesin. So we're putting one kinesin against 4 dynein because we're simulating for weak dynein, which is producing about 1.25 piconewtons of force. So 4 versus 1, force-wise, they're well matched up. But of course, there's a lot of dynein, so that ensemble is more robust. And so what happens, the actual locations of the motor heads come straight from the simulation and the cargo as well. And you can see that at high temperatures, dynein just wins because there's a lot of them. And they just win through sheer numbers. Now, as you lower the temperature, what happens is that this tug of war that you brought up becomes very vivid. You can see them struggling against each other. Kinesin is pulling one way, dynein the other way. It's really kind of cute. But at the end of the day, dynein still wins. Okay. But what happens when we go close to 0? Usually the buffers and the cytosol and all those things have salt, so you can actually go close to 0 and this thing will not freeze. And what happens when you go to really low temperatures is that you see kinesin here is active. And I invite you to look very carefully at what happens to these red dots here individually. And what you'll see is that they very rarely move towards the minus n to the left. They basically mostly just sit there. In other words, the activity from dynein, the individual dynein motors, their activity is basically negligible. All that's happening is that they sometimes bind and they sometimes unbind. That's the entire dynamics associated with dynein. Whereas kinesin is still moving reasonably okay. And so what's happening here, the reason why this cargo is moving to the plus end is not because kinesin is winning. It's not because kinesin is pulling the cargo and dynein is just detaching behind it and everything gives up. What's actually happening is that kinesin is biasing the location of the cargo a little bit towards the plus end. And so when the dynein individual molecules detach, they're more likely to reattach a little bit towards the plus end, under the cargo. And so that biases the binding forward. So really what we're dealing with here is a random on-off process, which is biased by a non-equilibrium contribution from kinesin. And of course, that's the textbook definition of a Brownian ratchet. So we have a whole new realization of a Brownian ratchet mechanism in this situation. So we were very excited to see that, because every time you see a new realization of a Brownian ratchet, it's kind of really interesting in and of itself. But here, it's very biologically and engineering-wise relevant, because the whole reason why we switch directions, why we're moving to the plus end, is because we switched the mode of motility. We went from power stroke driven, the motor is just going this direction and it's winning, to rectifying random motion. It was switched the mode, the entire physics has changed as we went to low temperatures, and that commensurate with that, the directionality of transport has changed. And that assures us that this effect is going to be very robust. There is a reason to look for it in real cells, is because this is a fundamental switch in behavior as you cross over below the temperature where dynein turns off. Yeah?
So is there a clear underlying physical reason for the turn off temperature? Does this correspond, for example, some excitation energy, some vibration or rotation of the molecule?
No, so the way this usually works is that dynein is a kind of a ring of domains which bind fuel, and one domain has to kind of collapse around the fuel, and then hydrolysis happens, and then it goes back, and it goes through this cycle. And the other domains also bind fuel, but they don't actually do the major motion which causes the whole thing to move, the whole motor to move. And what they do is they regulate the first domain. So their activity tends to basically tension the ring a little bit to change how the first domain is able to process ATP. And so what we're thinking is that the AAA3, the other major domain in that ring, it changes its behavior at below, let's say, 15 degrees. That's what we think is happening. So that's one story. And a lot of this work was done by a student in my lab, Willie Hong, who graduated about a couple of years ago at the end of 2014. And it was done in collaboration with Ambarish Kanwar at IIT Bombay and his lab. We are co-organizing a symposium in 2018 in IIT Bombay and trying to get support from the Biophysical Society as well. So the other story I want to tell you has to do with kind of zoomed out view, something that Saviz has already previewed, and that has to do with the overall transport complexity and the holographic trapping and all that stuff that he talked about. But let's start simple. Let's start with a picture I already shown you, which is the basic schematic of the cytoskeleton in cells. Again, you have the nucleus somewhere near the center, and then you have the microtubules that fan out from the logical center of the cell out to the periphery, and that helps support transport. And you can see here all these microtubules as they're fanning out. They're kind of nice and, I would say, laminar, if I'm thinking of flow. But this is what the real cytoskeleton looks like. So in the last about 10 years, we've gained the ability to look at the cytoskeleton. There's too much light. I don't think you can see all of them in their gory detail. Maybe that's better. Okay, I don't know. All right, fine. So you can see in gory detail what these filaments look like. There's nothing laminar about this. This is near the edge of the cell. So the edge of the cell is somewhere here. You can see that. And this is a three-dimensionally coded picture. So where you see stuff being more yellow, those microtubules are a little bit higher. And where stuff is more violet, those microtubules are a little bit lower. So you can see that they're not just crisscrossing each other in 2D. They're all over the place in 3D. And there are many places where microtubules come close together on the order of 100 nanometers or 200 nanometers. To give you an idea, what's relevant scale here is that a lot of the cargoes, viruses or whatever other cargoes, they're on the 100 nanometer scale. Some of them, like the nucleus, are on the micron scale. So when these things come together that close, that means that if a cargo has multiple motors on it, some of them can engage on one filament, the other ones can engage on the other filament. Now you have a whole different tug of war. So you were talking about tug of war on a single filament, but now think about what would happen if even the same motors, like kinesins. Some kinesins engage here, some kinesins engage there, and before too long you have a tug of war now between kinesins. And we've actually published a paper fairly recently, last year, on demonstrating that this type of tug of war actually happens in cells. This is another demonstration. So what I'm going to show you, let me just set it up. This is a COS7 cell, like a mammalian cell. And the microtubules are labeled green here. They're labeled with this protein called EB1, which primarily binds to the plus end. So you can actually see enhanced stuff here near the periphery. But it also binds along the filament, so you can actually see a little bit of the microtubule structure in a cell. And in addition to that, you probably can see those little bright dots all over the place. That's actually bleed through from the red channel. This is imaging viruses, in this case adenovirus, which has invaded this cell. There's a lot of virus particles in here. Saviz didn't think I would be interested in viruses. Well, this is something that we're very interested in. Now, I have highlighted one virus in red. Basically, what we have done is we have tracked all the viruses that were moving in this cell within a time window. And I've picked a few tracks to highlight for you what's actually happening. Because if you were to superimpose all of these tracks together, you would get this picture that I showed on my title slide, this really complex pattern of stuff moving around. But this is what happens to just a few particles. So you can see in blue is their track record, and then in red is where they are instantaneously. So you can see that this thing is not moving in any sort of a directed fashion. It's a DNA virus, so it tries to get to the nucleus nominally. And there are a few sitting next to the nucleus. This is the organizing center. But a lot of them are doing something pretty crazy. They're going all over the place. And you can see that they are exploring different filaments. They're switching from filament to filament. There's a lot of complexity to this motion. And we're interested in adenoviruses, herpes viruses. We're actually interested in virus motility because viruses are really nice prepackaged things that bind motor proteins and move and so they're very nice things to study in that sense. One of the things that we want to understand going forward, and I think, like Xavier's mentioned, this is the kind of complexity that's going to be really key for the field for the next at least 1 decade, but probably longer, is the fact that the cytoskeleton is actually very complicated. It's a three-dimensional structure. It has a lot of internal intersections. And if you want to understand what's going on, there's so much complexity. In addition to that, there's also regulatory complexity. There's a lot of proteins that modulate what's going on. So if you want to understand it from the cellular side, it's breaking apart what happens in cells, as biologists usually do, you'll have a very hard time coming up with a quantitative description, modeling what's actually going on. So if we want to understand cleanly what's going on, we'll have to build it from building blocks, from simple building blocks, and we're going to have to watch out for emergent complexity. And so if you want to build 3D cytoskeleton, then that's a very challenging thing because you need to be able to manipulate filaments that are 25 nanometers across. They're barely visible optically, so very hard to work with. And what we want ultimately, I wrote here flexible and efficient assembly process. What I mean by that is that when you work in vitro, in order to learn the rules of the game, you have to set up pretty much the same initial conditions and let the system go through the entire experiment, let the cargo navigate through the intersection and see the outcome. And you need to build statistics. So you need to be able to do this many, many times over. You need to do this very efficiently so that you can make meaningful statements about whether there are any statistically significant differences. And in addition, of course, the goal eventually is to build a cell. I mean, I'm not kidding. The goal is to build a cytoskeleton, to put as many components as we can put in there and to model as much complexity as we can fit into a glass slide. And that's a lot of complexity in principle. But we need to have an assay that allows us to add this complexity with an eye towards the future. Now, the other motivation for us in exploring 3D and these filaments is something that probably a lot of people in the audience are familiar with. There's a famous manifesto from Feynman, There is Plenty of Room at the Bottom, where he first laid out in the 50s the vision of nanotechnology in a slightly more abstract way. And he described it it was electron microscopy centered because that was the big thing at that time. But in the early 80s, you have Eric Drexler coming out with a famous manifesto of his own, Engines of Creation, where he describes what we now has now morphed into our vision of nanotechnology. The idea that you'll have machines which build things and manipulate things with atomic precision, and you can build things on a nanoscale. And he already realized at that time that if you're going to have that happening, here and there, you're going to need, just like in a cell, you're going to need to be able to efficiently connect those areas. You're going to have what he called conveyor belts to connect these nanomachines. And so, not surprisingly, we're very excited about microtubule system because not only are these nano conveyor belts potentially, but they come with a built-in machinery from, associated machinery from cells which move stuff along. So it's a really exciting system in that sense. And by the way, we're not the first ones to notice that microtubules are really great for moving stuff around. There's a ton of research on the bioengineering side, and trying to adapt microtubules for engineering purposes. People are trying to build interesting shapes out of microtubules, like spools and asters, and people are trying to use microtubules as trains for moving stuff. So if you attach motors to a glass slide and then put a microtubule on top, the motors will start moving along a microtubule, or alternatively, the microtubule will start moving relative to the motors. And so you'll have microtubules gliding along the surface. And if you then attach other stuff to microtubules, then you basically have trains carrying interesting stuff from point A to point B. And you can route these things by etching channels on the surface to route these microtubules, because when they bump into an obstacle, they will then bend. And so there's a lot of research on that. Alternatively, there are proposals to use microtubules, like Cell does, to sort of direct transport of cargoes using molecular motors. The problem here, of course, is that you need to orient microtubules. You need to control their polarity. You need to connect to point A to point B and not point B to point A. You want to know that it goes from here to there, not from there to here. And so a lot of these proposals have efficiency issues where 90% will be oriented correctly and some will not, and that's a problem. So ideally what we want for all the applications, biological and engineering applications, what we want is to be able to work with individual microtubules. That's the ultimate miniaturization of technology. And we want to be able to control the polarity. We want to know where things are going, where we expect things to go. And then we also need to be able to position things in 3D. Those are big goals. And so our vision when we started was to attach refractive particles to microtubule filaments. And filaments themselves are very hard to manipulate, but refractive beads are very easy to manipulate using techniques like optical trapping. And so let me briefly mention to you how optical trapping works, is that you basically have an objective and you inject a laser beam into the objective, and it focuses the laser beam. And if the numerical aperture of the objective is really high, then it focuses the laser beam really tightly. And it turns out that if you put a refractive particle at the focal point of this laser, then if it tries to get out, it will feel a restoring force in all three directions. And so basically, if this cargo is just feeling the Brownian kicks, then it's pretty easy to trap. But if it has, for example, a motor and you bring it next to a microtubule, it will engage on the microtubule, and then that motor will try to pull the cargo out of the trap. And now you can study how this motor works, but ultimately you need a stronger trap for that. So this type of a technology is great for, of course, positioning things. It's great for examining the chemistry at the single molecule level. You can figure out whether the motor is moving, how fast it's moving, how reliably it's moving, how much force it's producing. All of that is accessible with optical trapping. And it's great because if you wanted to reposition this trap, move it somewhere else, or timeshare it, create multiple traps by quickly going from one place to another, you could do that. You can move traps really fast, up to about 100 kilohertz is pretty easy to do.
How can you determine the direction of the move?
The direction of what?
The traps.
Oh, so all you have to do for the traps is to rotate the laser beam in the plane that's conjugate to the back focal plane of the objective. And then the rotation in the back focal plane of the objective translates into translation in the image plane. So all you have to do is have something that rotates the beam. And you can use a mirror, the simplest, or you can use acousto-optic deflector or electro-optic deflector. There are many technologies that allow you to do that. AODs go 100 kilohertz standard. That's a pretty standard technology. But the problem is that all of this is inherently limited to the plane of the objective. It's not 3D. Because the whole point of this is you have an objective and it's focusing the light. So you're always going to create the trap right at the plane, the focal plane of the objective. The alternative is to use what's known as holographic trapping, which is Savis already mentioned. And this is probably one of the simplest explanations for it. The idea behind holography is that you can configure the electromagnetic fields in the volume if you can control the boundary conditions. So here, imagine shining two laser beams into the objective at a slight angle. Then basically you're going to create two optical traps slightly off axis in the focal plane. But the key thing is that you're going to create some sort of an interference pattern between these two beams that you're injecting in the back focal plane of the objective. Now, imagine if you were to shine just a single laser beam, but you had some sort of a machine, we'll call it a spatial light modulator, which could impose this boundary condition right here so that the field would look the same in the back focal plane of the objective. In that case, the propagating light through the objective and outward will not know what happened before this boundary condition, and it will recreate the same two traps. So essentially, by controlling the boundary conditions for a laser beam at one particular location, you can control then what happens, what your light looks like in the sample. And in fact, these optical traps that you can create with holographic technology have a lot of the same benefits as the ordinary traps. You can still move stuff and reposition stuff anywhere in the field of view. You can look at single molecule behavior, gives you the same access. The problem is that these cannot be moved fast. The spatial light modulators that are available go about 100 hertz. 100 hertz, not kilohertz. So this is really slow technology. But turns out that you can actually design the boundary conditions so that you can make traps not only in the focal plane of the objective, but in 3D. So you have two technologies which are related, of course, but these are two technologies which have complementary strengths. And so we thought, well, let's combine them. And we were the first ones to do that. So this is a development that's unique to Utah. We have a very unique machine here, and we're almost done building a second one, where basically you have one line, laser line, going through a spatial light modulator and creating a holographic trap, so we can build things slowly but in 3D. And then we have another laser line, which is an ordinary line, so there's only one trap. We don't have multiple traps. We don't have 3D with that, but we can move it really fast. And so if we wanted to probe what's happening, if we wanted to respond to what's happening in our system, we could do that on the scale of many kilohertz. This is what the system looks like. This is kind of an older picture. This system keeps evolving and getting fancier. So one line here is the regular optical trap, and the other one shown in green here is the holographic trap. You can see it bounces into the spatial light modulator, which is in the conjugate plane to the objective, which is sitting somewhere there. So the student who did a lot of this work and is responsible for a lot of our recent advances is Jared. He's a student in biology. He joined my lab like 3 years ago. And this is kind of the before picture. And this was a hard project, so this is the after picture. But this is what he did. So we needed to basically be able to hook up beads to microtubules reliably. So the first thing is, how do you hook up beads to microtubules? So we took inspiration from nature. We took Kinesin motors, and they bind really nicely to microtubules, and we can make them. That's fine. But of course, if you give them fuel, they'll start walking. So that's not very good for manipulating microtubules if your handle starts moving along. So we didn't like that a whole lot. So we engineered these motors. We made a mutation, a single point mutation, which turns off enzymatic activity. So these motors just bind and sit there, and they don't go anywhere. So they're really good glue, basically, that gets the beads stuck on microtubules. Great. Now you have another problem, which was actually vexing to us for a while, which was that if you make beads which are very sticky to filaments and you mix them together, and you have to do that, then you're going to end up with a mess. You're going to have beads which have a lot of microtubules on them, and it's just going to look like a porcupine, and you have no control. And the way we solved that problem is that these beads, we started using beads which are made of glass, which is 2.2 times the density of water. And so if you mix them at low concentration with microtubules and you quickly put them into a flow chamber, they sink really fast. And so now you have beads and microtubules. Microtubules float. The beads are at the bottom. They don't necessarily talk to each other. And then you can basically take a bead, pick it up. You have to passivate the surface really nicely so nothing sticks. But you can take the bead, pick it up, put it on the microtubule, and now you have the ability to assemble the cytoskeleton. Now you have everything working. So let me show you a few things here. First of all, I don't know if you can see it very easily, but there's a bead here, and there's a microtubule that's sticking out of it. Can everybody see that? Awesome. So this actually happens. Well, these are pretty hard to see without some additional processing. I'll show some videos where we've done some additional background subtraction where they kind of jump out at you. But you know, with just bright field illumination, they're a little bit hard to see. So it's good to double check. So this bead in particular already has a microtubule on it, and it's just sitting on a glass slide. And this black dots here, they just denote the position. There's no special importance to it other than a fiduciary for where this bead was. And there are fiduciary markers on the surface to tell you that we're not moving the surface. And then there's another bead which has nothing on it. It's just sticky. It has these dead kinesins on it. But these red circles denote the fact that we're optically trapping it. So we can actually manipulate this bead. We can bring it next to a microtubule. It will immediately bind to the microtubule, and you form what we call a dumbbell, where you have two beads and a microtubule in between, exactly the kind of building block that we're looking for. And then we can, for example, drag one bead and the other one follows, so you can see that this connection is made really well. Yeah?
What causes the microtubules to actually bond to the glass beads? You wouldn't think that they would be bonding, really.
Right. So what we do is we chemically functionalize the silica surface, the glass surface. We silanize it, and then we use various attachment techniques to the tags on the motor to hook it up to dead kinesin motors, which are enzymatically dead. They're not moving, but they still retain the ability to bind to microtubules. So we use a biomimetic approach. We use motors which bind to microtubules as the glue, and then we figure out how to attach those motors to the beads. So, and it's actually very nice because we use full-length motors, so there's a 100 nanometer long thing which is very flexible. So they're exploring all the space around them, and if you bring this bead next to a microtubule, it readily binds. It immediately finds it and it just binds. If you use something like an antibody, which is only a few nanometers, then you have to really kind of press it in, and there are other problems with antibodies. But the point is that this is much more efficient and much more convenient, and if we're looking for high throughput, this is the way to go. Okay. Now, if you can make one dumbbell, you can make several. And what you can do then, this one was processed differently, so you can see microtubules a lot easier. But so what we did here is basically you take two frames, a few frames apart, and you subtract one from the other. So all the background disappears, and the microtubules kind of jump out because they vibrate. So when you subtract, they're not always in the same location. So the image of microtubules becomes really vivid. So you can see here dumbbells with individual microtubules hooked up between these beads, and they're all lying on the glass. And we know exactly where they're lying, so we can make a depot of these things as building blocks. And then, if you want to do it industrially, especially if you want to automate it eventually, then basically you can make this in quantity. And then what you can do is you can build stuff out of them. So let me show you this video. So again, it's going to be a little hard to see until the video starts. There is a dumbbell here, there's another dumbbell here, and there's a beads attached to this dumbbell in the middle. And there are two very long dumbbells, probably about 20 micron long microtubules here that are sitting here. So there are 4 dumbbells here in this picture. So this was accelerated 6 times, and you can see. So Jared lifted one set of beads up, snuck the other dumbbell underneath, made a cross, and now he's manipulating this cross to make it into a pyramid. So this central bead will be sticking up, so it's basically a 3-dimensional shape, and now he has that. He's manipulating it, and then he's cross-linking the base with these long microtubules so that it becomes a stable structure. Once he has completed it, at the last frame, you're going to see he releases the optical traps, and this thing just goes. There we go. And it just goes as one shape. So you can actually build 3-dimensional shapes out of microtubules. Again, I invite you to think about the scale here. We're manipulating 25 nanometer wires here, basically, and we're building stuff out of them with essentially nanometer precision. You can use other building blocks. You don't have to use dumbbells. Here's a pentagon and a Y shape, and you can manipulate them together. Okay. What happened? Okay, somehow it's gone a little too far ahead. Yeah. So I don't know why it's done that, but all right. So you can start with these building blocks and then combine them together to make something that's pretty complicated. And the point of this structure is basically, there are a lot of these technologies like DNA origami and so forth, which allow you to build fairly complex things, but nothing like this. This is a very high level of complexity, and again, we can do this in 3D, and we can do this on demand pretty much immediately. And again, we're thinking about automating this to make something like a 3D print, not a printer, but a 3D construction thing where you can say, I want this shape, and it says, okay, here are the microtubules, here are the beads, let's build it. Right now, students do this, and it's a vision. It's something we're working on, but it's a very appealing vision. And the other thing is that, something that I really highlighted before is using microtubules as tracks to connect different areas, it may be in your nano device. And here you can see, if you think of these as proxies for different areas, you can see we can lay down tracks as we please. So this was built as a kind of a extend. This microtubule is very long. Some of these are shorter. This is probably like 30 microns long. So these are very extended, large-scale constructions with individual microtubules. Yeah?
Michael, are the length of the microtubules, is that something you can control directly, or do you rely on kind of a distribution?
So there are two answers to that question. First of all, there are papers out there, and we certainly explored that a little bit, on how to control the length of microtubules. Ultimately, the simplest thing is to incubate things for a little bit longer, give it a little bit more material, and you'll get longer microtubules. That's the short answer. There are other approaches. You can take microtubules, kind of pellet them, and use them as seeds to grow longer microtubules later. So bottom line is you can make microtubules pretty long. Up to 100 micron is doable. But the other answer to that question is that when you put them in your slide, you are the one who goes with that bead and hunts for a microtubule to attach it to. So even though there's a distribution of length, you can pick microtubule that you want. So in some sense, you have more control than just with random incubation that would give you some sort of a overall distribution with maybe an exponential tail. But you can find very long ones. That's not a problem.
Do the glass beads actually, can they shorten a microtubule? Can they attach it at the middle and break it? Or do they always go to the ends of the existing microtubules?
No, the glass beads don't really break it. But if you flow very long microtubules through a small opening, like let's say if you don't cut your pipette tip when you do it, then you can shear microtubules. So microtubules can be sheared, but not under those conditions.
What's the time scale of the process?
Well, by hand, the time scale is a few hours. So, but the nice thing is that once you build it, you can build it on demand, and you can basically use it for whatever your nefarious purpose is. So let me show you one nefarious purpose, which is to study transport. Like I mentioned, one of the key things in cells is how do cargos navigate all of these intersections between cells, because the cytoskeleton is such a mess in a typical cell. So if you want to do it cleanly, you want to start at the level of a single intersection. And so these are bead handles. They have dead kinesin. They hold 2 microtubules. This one has live kinesin on it, several motors on it. And you can see it's moving. There's no glass slide. It's all in 3D. It's moving along this filament. It comes to the intersection. It engages on the other microtubule, and then it snaps, and it keeps going. So you can study the outcomes, the statistics of the outcomes directly. In this case, this microtubule is 500 nanometers above the other one. This one is actually fairly visible because it's in focus. The other one is above focus, so it's hard to see. It's 500 nanometers above the other one. So we can control the separation, and we can do these measurements as a function of angle, as a function of distance. We have a lot of control. So Saviz was presenting a vision, but he wasn't exaggerating that far. We really can build these things on demand. And if you think about the grand vision here, at least as far as transport across many intersections, one intersection is nice. You figure out the rules, and then you have some sort of a model in your computer that tells you what's happening. But the question is, does it scale? Because there is always some error bars to your statistics. And the question is it going to scale? And so what you're going to have to do later, once everything is figured out, is to make 2, 3 more intersections and see that the model scales up. And once you show that it scales for several levels of intersections, it's basically a Markov process, right? You make choice at one intersection, you go for a while, come to the next intersection. So in that sense, you can think of it as stacking one choice on top of another. And basically, if you show that this stacking statistically works, that everything scales nicely, then that gives you then an opportunity to think about what happens in a cell. Because again, super resolution today gives us perfect ability to see these filaments where they are in 3D. So we can figure out what the layout of the cytoskeleton is in 3D, and then we can figure out what the cargos would be doing at these intersections, and then we can start predicting what the distribution of cargos would be in the entire cell from first principles, essentially. So that's a very powerful vision. Of course, we don't expect that clean microtubules with just kinesin-driven cargos will mimic what happens in the cell, because there's a lot more complexity in the cell. So eventually, this will give us a window to study what else is regulating transport. That's kind of the grand where this is going. So the conclusion is that this stuff works, this stuff is flexible, and this stuff is really exciting. What we are trying to do, again, is to work on better visualization, and we're trying to figure out how to automate this process. This is kind of in the back of our minds at all times. So let me talk a little bit about the future directions now. So the first thing is concerning this 3D transport. We obviously want to understand how cargoes choose which way to go, depending upon how many motors they have on these cargoes, and the spacing and the angle of these intersections. So we're trying to map out the phase space really well. And if somebody is interested in our recent data, which we're preparing for publication, I'm going to give a talk in math biology on September 21st, so a couple of weeks from now, where I'm going to go into the data that we have with this system. Well, we have quite a bit of data. But let me say one thing about one piece of data that's most relevant to this talk. And that is, if you start with intersection that is at 90 degrees, and you have microtubules that are 0 nanometer separation, so they're basically touching, versus 500 nanometer. And for scale reference, the cargo we're using is 1 micron size. So basically, you're either undercutting the cargo or you're right hitting it down the middle. So that's the scale. And you can see that in one case, if there's 0 separation, then the preference is to switch. Whereas if there's a separation of 500 nanometers, the preference is to keep going on the same microtubule, and the difference is significant. And we actually have more data for like 1,000 nanometers and for different angles, not just for 90 degrees. So we have a lot of data on this. And we actually think we have mapped out, at least for this condition, for those cargoes, what the phase diagram basically looks like. But again, I'll talk about it in 2 weeks. And we're working with Ambarish to simulate this as we speak. So, but the conclusion from here is twofold. First of all, this is direct proof that demonstrated from the data that says that the transport is inherently three-dimensional. You're never going to learn how things move in real cells by sticking microtubules on a glass slide. You have to do it in 3D. You have to do it probably with this technique, or I struggle to think of an alternative, really. And the other thing that this shows is that cells, by just changing the spacing between filaments, have the capacity to regulate whether things switch or pass. In other words, they can move, navigate cargoes as they please by just changing the spacing between filaments. That's in principle available to cells as a possibility. So that's very interesting from a biological perspective. Now, the other thing that we recently got funded from NSF to do is to look at the mechanics of microtubules. So let me very briefly touch upon that. It also uses our 3D construction technique. So this is an old experiment from Florin Lab at UT Austin, where they basically grafted microtubules on some pillars, which were 3D micromachined on a glass slide. And then they attached a fiduciary bead, and they just basically looked at how these microtubules were flopping around due to thermal fluctuations. And what they found was something very interesting. What they found is that the persistence length is pretty low for short, well, low. It's millimeter. So it's a very long, if you think about something that's 25 nanometers across, right, the persistence length over which this thing looks like a rigid rod is like a millimeter. That's pretty amazing, but that's pretty short compared to what happens when these filaments get really long on the scale of, let's say, 40 micron. In that case, the persistence length climbs 5 to 6 times. And the crossover point is somewhere around 20 micron. Okay? And the reason for this unusual behavior, so this isn't your usual Euler beam. The reason for this is that this structure is actually highly anisotropic. And it's this anisotropy and the difference between the shear moduli in different directions, that's what gives you this behavior. But let's not even get into that far. Let's just think of it as physicists often do. What this plot says is that microtubules have a built-in length scale. There's a length scale at which their mechanical properties undergo a crossover. And that length scale is about 20 micron, which happens to be close to the length scale on which a lot of eukaryotic cells exist. And so let me skip this slide. I think I'm running out of time. And so the interesting question that arises is whether or not this built-in scale for a single filament implies that the cytoskeleton as a whole has crossover behavior at higher scales. And so we want to probe the mechanical properties of identical structures at different scales to see whether we pick up signatures of changes in mechanics and how microtubules might contribute to cell shape maintenance at different scales. And this is one of the places where having the ability to move a trap really fast, not just build things in 3D, but also to move things really fast is key. Because you want to probe the moduli across a wide range of frequencies. You want to go from very low, a few hertz, to a few kilohertz at least. So you want to be able to modulate these beads, basically, impose some sort of a stress on them directly. Okay, and you want to do it at different frequencies up to a few kilohertz. So our machine is precisely built for these types of experiments. And the other thing, of course, is that if you think of the cytoskeleton in the simplest case where these microtubules just fan out, then you will never encounter this problem. But if you think of a microtubule as a jumbled mess near the periphery, then you have to wonder what the mechanical properties are doing when you put in the cargos. Because the cargos, in the process of having tug of war at intersections, they also cross-link filaments. And that has to have an effect on the mechanical properties of the entire network. And so we're very interested in using our beads with different attachment chemistries as different cross-links to study what are the mechanical properties of the entire network. For context, this is something that comes up a lot for actin networks, another cytoskeleton filament. Different cross-links give you completely different behaviors mechanically for these types of networks. So, but the problem is that actin networks can form a hydrogel, so they're very easy to work with. Until we developed our technology, microtubules were hard to work with because they were long filaments and they're pretty sparse. So you need to build structures with some control. We can do it, but you're not going to do it by just throwing microtubules into a glass slide. So we're the first ones who can address the question of what happens to microtubule networks as you change the properties of crosslinks, and microtubules themselves. We can modulate that too. So that's kind of an example of what we're going for. And the last slide, I just wanted to give a shout out to members of my lab. So this is Jared, who is working a lot on this 3D stuff. And Flo just joined the lab working on similar type projects. Olu is working a lot on this temperature stuff that I mentioned. Again, we're trying to establish temperature as a good hammer to probe what happens in cells and in vitro systems, to learn about the dynamics of multiple motors and the different modes of motility that can give you. So there's a lot of exciting stuff there. And I wanted to thank the funding from Utah Research Foundation and from the NSF. So thank you. Sorry, I guess I ran over a little bit, but if you have any questions, please. Oh, yeah.
Beads afterwards, like somehow.
Yeah. So.
Induced photochemistry that turned the cross-linkers off and gave the beads out after you have built the structure.
Actually, it's easier to do on the other end, and at how you attach the beads to the motors. So you can make those connections in a reversible fashion, so you can then basically detach the motors from the beads. So we've thought about it. We haven't done it yet. We've definitely given it a lot of thought about how to do that, but we haven't done it.
You can replace it with, you can throw in smaller beads or smaller cross-links and then just cross-link the actual microtubules.
Yeah, well, I mean, there are some things that are easier to do this way and some things that are harder. So, for example, exchanging just one cross-link in a network would be hard. But, exchanging a lot of them would be fairly easy. And the more control you want, the harder it gets in that sense, once you've built it. But if you don't cross-link things, if you hold microtubules in place without cross-linking, like when crosses, there you hold the bead handles without actually them cross-linking microtubules, so it's fully dynamic. Then you can reconfigure it at will. That's a no-brainer. Yeah.
So you said the experiment was anisotropic. That means like independent of or dependent on the reference frame, right? What did you mean by that?
No, what I meant was anisotropic means that what happens to this filament depends on which direction basically you impose shear strain. So the way this works is the following. So imagine so the building block is actually alpha and beta tubulin dimer. So it's kind of a little dumbbell, and then you're sticking one after another to make what's known as a protofilament. So these are very long filaments, alpha beta alpha beta alpha beta, kind of a very long structure. Let me actually draw it. Okay. So what's actually happening, you know what? I might have a slide on this. There. So your building block here is alpha beta tubulin dimer. So it's a kind of a blob. And then you're sticking one after another to make what's known as a protofilament, a kind of a linear structure. And then you're binding them sideways to make a tubule. Okay? Now here's the key. Here's where the anisotropy comes in, is that the bonds that are along the protofilament are pretty strong, whereas the bonds on the side are made with flexible loops, and they're actually pretty weak. So you can think of this as an assembly of sticks which are very loosely bound. So basically the connection along the microtubule is different than the connection circumferentially. So it's a very anisotropic structure. And so what happens actually is if the microtubule is short and you try to bend it, then these weak connections side to side don't talk to each other much. And you're just basically you're basically bending 13 protofilament at once, but individually, essentially. They don't talk to each other much. If the microtubule gets particularly long, then all of these weak connections add up. And now there's enough overall interaction that when you're trying to bend the microtubule, what happens is that you're trying to bend those protofilaments, but they're now together in a bundle. And that's a lot harder to do. Basically, they don't want to slide relative to each other, which they would have to do if you wanted to bend it. So there is the crossover, and that crossover happens at about 20 micron. Yeah?
So do you ever change the size of your little glass balls and see what, they're huge compared to the normal cargo, right?
Yeah, so first of all, they're not. I mean, if you look at nuclei, for example, as the biggest, then some of the biggest beads that we use are on the same scale. But you can go down to about 500 nanometers, which is fairly reasonable. It's not going to be like viruses, but it's fairly reasonable for a lot of the intracellular cargoes.
You don't think that if the size of the bead doesn't affect the behavior of the motors and stuff like that too much?
The behavior of the motors, no. But one of the things that we're interested in is how the properties of crosslinks will modulate the properties of the entire network. And there, the geometry comes into play a little bit, because basically the curvature of the bead will determine how much of the surface is visible from how many protofilaments, which means how well, if you have multiple motors, where can they bind, and that might actually have a little bit of an effect. We don't expect it to be a major difference, but we expect the major difference to come from actually changing the chemicals, the crosslinks themselves.
These beads is more or less covered with
but we can control the density.
motors, only a few of which are actually attached.
But we can control the density there. We're in control of what goes on the beads. All of that is, we have control over that part of the system. Yeah.
So in what sense is the transport deterministic? I think the purpose of the microtubule is the same, is to do something better than diffusion, at least.
Yeah.
But is there a directionality to put the cargo on the right railroad track, so to speak, so that it gets to the right location, even with these cross-linking?
Well, so this is a loaded question, right? So for some cargoes, it's really essential to go from Golgi to ER or something, or from ER to Golgi or something like that. So you want to connect those pretty well. In some cases, like for the adenovirus, seems to be wandering around. So it depends on the cargo in some ways, and that in turn probably depends on which motors it's sitting on, and then which adapters are also being used, because the adapters have a lot of role in modulating the properties of the motors, especially for dynein. So it's a very loaded question. There's a lot of variety in biology in general, but we're starting from a spherical cow approximation. And we're trying to walk away from it and gradually build the complexity. That's the whole point. So we will look at, there's certainly cargoes that simply have a bunch of motors on them and they just move, like adenovirus seems to be. They have several binding spots for kinesin and dynein motors, and it seems to have no problem engaging on different microtubules. And that leads to very complex behavior. That's the kind of stuff that we're very interested in understanding.
This switching that you're going to talk about in a couple of weeks, does that depend on, or how does it depend on the density of motors around the beads, or how's that? Can you quantify that?
Yeah, so first of all, if you just have a cargo with one motor, just one motor on it, you're not going to have a tug of war. Because this motor, it has two heads, so it kind of moves in a step-by-step manner, but those two heads cannot reach farther than 8 nanometers. So they're basically only seeing the next binding site. So they're not going to be able to see an intersecting microtubule. The only way you're going to switch from one to another is to detach fully and then to reattach. But as soon as you start having multiple motors, then you start seeing switching. And what we have shown previously when I was a postdoc for microtubules that are lying on a glass slide, so we didn't have 3D back then. We showed that if you increase the number of motors to some high limit, beyond like 3, if you have a kind of like ancients used to count, 1, 2, 3, a lot. But if you have a lot of motors on your beads that are instantaneously driving your beads, then basically a lot of the cargoes tend to get stuck at intersections. They get there and they just keep fighting for a while. What's a while? I mean, that's the other interesting thing is that people in the field tend to use 1 second as a cutoff. Like if this thing sticks at the intersection for a second, it's stuck. Whereas in reality, yes, a second is a kind of irrelevant time scale for a cell, but there are a lot of things which can wait for a second. It's not such a long time scale. So there's actually quite a lot of need to be more quantitative about it and not just use a simple cutoff. But there's certainly a lot of evidence that as you increase the number of motors, things start fighting for longer at intersections. And so conversely, if you put something like tau protein on microtubules, which tends to inhibit motor activity and kind of doesn't let them rebind very well, so you have net on the same intersection basically, but you have fewer motors, then you will have more efficient switching. And
that's a mechanism that the cell uses to regulate transport.
That's what we propose, and Jenny Ross, when she was in Ergo Holzbauer lab, they basically proposed something similar. So we had a converging view of this. Yeah.
So when you mentioned yeast, that made me think about plants. Yeast is not really a plant. But I was wondering to what extent the properties like dynein, kinesin, properties that you described in the early part of the thing, would also be exhibited in plant cells.
So that's a very interesting question. First of all, we're trying to look at plant kinesins, but the way plants organize things is very different. The organization of microtubules is very different. So this was for animal cells. In plants, microtubules are more parallel to each other. They don't form this array that fans out. And also in plants, the transport is mostly, at least in a lot of plants, is driven by kinesins. It turns out that there's a whole class of kinesins that can move towards the minus end, and the plants use those for a lot of their minus end transport. And so it's actually a very interesting question. If you can survive on minus end directed kinesins, why do you need dynein, right? What is dynein? And dynein is a really fascinating thing. It's 1.2 it's a huge thing. It's 1.2 megadalton. It's much bigger than kinesin, about 10 times bigger than kinesin. And it typically, in order to work, it needs another cofactor called dynactin, which is about another 1 point something megadalton. So that you have this huge thing sitting next to this puny little kinesin, and they're fighting each other. And it's really hard to envision why this is how it evolved. And plants give us a counterexample where it's mostly done with kinesins. And so it's a very interesting comparison. But there, you mostly have kinesins.
Something like slime mold, which is kind of on the border.
Dicty, yeah.
Plant and an animal. How does you know what the slime mold behavior is?
But it also has a slightly different, like more rarefied organization of microtubules. So again, it's a slightly different system. But yeah, I mean, it's actually an interesting system. You do have dynein versus kinesin issues there. So it might be an interesting thing to look at. And certainly one of the postdocs, a good friend of mine who's in India, another Roop Malik, he's working on Dicty a lot. So in principle, we could look at that system, but it's certainly not. So the reason why yeast is so important for dynein research is because it's the one place where dynein is non-essential. In other words, you can play around with it, mutate it, do whatever, and yeast is still alive. If you do that to us or to any animal, if you start playing around with dynein, it's at the center of everything, and it's lethal. But in yeast, it's not. And that gave the field a playground. And certainly, Vail Lab has made tremendous progress with that system. But also yeast dynein is different from mammalian dynein. There are important differences which have been proven and established over the years, and it's been a very interesting comparison between the two. Why they have different force production properties, why they have different velocities. Yeast dynein is very slow. It's like 80 nanometers per second. Mammalian dynein is like 1,000, at room temperature, 1,000 nanometers per second. So order of magnitude difference. And the question is why. And we think we understand there's a C-terminus domain in that ring which actually seems to modulate a lot of that. So there's some agreement on where things are coming from. And again, I sort of cover a lot of these details in a class that I teach. So I'm probably going to go to big ads. And if you want hours more of discussion, I would love to just keep talking about it.